Hello friends, today in this video we are going to discuss about the peptide, polypeptide and peptide bonds of amino acid. Okay, so the first thing is the peptide. So what is peptide? So peptide is a compound that contains at least two amino acid or more than two amino acid which are linked to each other by peptide bonds. Okay. So this is the peptide, and the peptide which contain more than 12 and less than 20 amino acid, that peptide is known as oligopeptide. Okay, and the peptide chain which contain actually more than 50 amino acid, that linear peptide chain is known as polypeptide, which are linked to each other. All amino acid, all 50 or more than amino acid are linked each other by peptide bonds. Okay. So these are the peptide, oligopeptide and polypeptide, and now we will see how the peptide bond formed. So this one is the first amino acid, one amino acid, this is another amino acid, and we will see the peptide bond formation between these two amino acid. So here the peptide bond can be formed by the alpha carboxyl group and the alpha NH2 group. The peptide bond can only be formed between the alpha carboxyl group and alpha NH2 group of another amino acid. Okay, so here alpha means, what is the means of alpha? What is the meaning of alpha? Alpha means this functional group is attached to the alpha carbon. So this one is the alpha carbon, and what is the meaning of alpha carbon? Alpha carbon means the first carbon which is attached to the functional group. So this one is the functional group, so it is attached to the first carbon to which it is attached. So this one is the alpha carbon. So for this functional group, the NH2 group, this one is also the alpha carbon because it is the first carbon to which this functional group is attached. So this one is the alpha carbon. So these are the alpha carboxyl group, and this one is the alpha NH2 group. So the peptide bond will be formed between alpha carboxyl group of one amino acid and the alpha NH2 group of another amino acid. So now we will see the OH group of carboxyl group and one hydrogen of NH2 group of another amino acid will release as a water molecule, and now we will see a peptide bond formation between these two amino acids like this bond, that is between the CO and NH group, that bond is known as the peptide bond. Okay, and now this compound is known as the dipeptide. So now if another amino acid will come and join to it, we have to join it, then the NH2 group of that amino acid will attach to this, react with this OH group, and this OH and the hydrogen of the NH2 group will be released as a water molecule, and they form a peptide bond between them, and that will be called the tripeptide. Okay, so in this way many amino acid can be joined to each other and form a polypeptide. Okay, so here one thing is that the first amino acid, their free NH2 group will be present, that will be known as a N terminal, and the last amino acid of the polypeptide contain a free COOH group, that last amino acid, that end is known as the C terminal. So the first NH2 group will be free, that is known as the N terminal, and last COOH group is free, that is known as the C terminal of that polypeptide. Okay, and here one thing is about the peptide bond, that is this peptide bond has a partial double bond character. So here we can see there is a partial double bond character of this peptide bond because of resonance, because this structure, this double bond can change the position, because here a delta minus and here is a delta plus on the nitrogen and delta minus on the oxygen, and here a partial double bond character is present, and here the O minus and O plus. So here a equilibrium of the three structure are present in the nature. So here a partial bond structure is present because of resonance. So because of this character, this peptide bond is a rigid planar structure. Because of this partial double bond character due to resonance, this peptide bond has a rigid planar configuration. Okay, so this is all about the peptide, polypeptide and peptide bonds. Now we will see the calculation for molecular weight, calculation of molecular weight for protein and amino acid. How we can calculate the molecular weight of protein by counting the amino acids or peptide bond formation. Okay, so the first numerical is, if an amino acid have molecular weight of 75 Dalton, then what will be the molecular weight of dipeptide and pentapeptide of the same amino acid? That means same amino acid of 75 Dalton of molecular weight, and if they form a dipeptide, then what will be the molecular weight of that dipeptide? So here we see this one is the amino acid, that molecular weight is 75. This one is the same another amino acid, that is also the molecular weight 75 Dalton. So when they form a peptide bond, after that we will get a 75 into 2, that is the total molecular weight. But due to peptide bond formation, here we can see that due to peptide bond formation, a water molecule is released. So we have to subtract the molecular weight of water, that is 18 Dalton. So here 75 into 2 minus 18. Because of peptide bond formation, this dipeptide have to lose a water molecule. So for that we will get this Dalton, this molecular weight. Okay, so individually they have total 150 Dalton, but when their dipeptide form a peptide bond, their molecular weight will be less by 18. Okay, so this is the molecular weight of dipeptide of the same amino acid. Now we will see the pentapeptide, molecular weight of the same amino acid of pentapeptide. So here we see a one rule, that is for 2 amino acid the peptide bonds are 1. Here we can see for 2 amino acid the peptide bond is 1. Okay, so 1 water molecule will be released. So for 5 amino acids, suppose for 5 amino acid, here we can see that the amino acids, 5 amino acids are linked by the peptide bonds. So there, how many peptide bonds will be there? So 1, 2, 3, 4. So there will be 4 peptide bonds for 5 amino acid. So here from this we can get a formula that for n number of amino acid, if in a peptide n number of amino acids are there, then the peptide bonds will be n minus 1. Total peptide bonds will be n minus 1. Suppose if the 5 amino acids are there in the peptide, then the peptide bonds will be n minus 1, that is 5 minus 1, that is 4. So for pentapeptide, there are 4, total 5 amino acids are there. So 5 into 75, because 75 is the molecular weight of one amino acid. So 5 into 75 minus how many peptide bonds are there? There is 4. So 4 water molecule will be released. So 4 into 18. So here is our molecular weight of the pentapeptide of the same amino acids. Okay. So now we will see the next question, that is if a protein have a 100 amino acid residue, then calculate the molecular weight of that protein. Suppose a protein contain 100 amino acid. Okay. So now one thing we have to remember, that the average molecular weight of an amino acid is 128 Dalton. But remember that the average molecular weight of amino acid in protein is 128 minus 18, that is 110 Dalton, because in a protein the amino acids are present by their peptide bonds. They link to each other by peptide bonds, and due to peptide bond formation, one water molecule is released. So this is the average molecular weight of an individual amino acid, and in a protein the molecular weight of amino acid will be less by 18. so that will be 110. So the molecular weight of an amino acid in protein, that is 110. So now our total amino acid is 100. So 100 into 110. So for 100 amino acid, how many peptide bonds are there? That is n minus 1. So that is 100 minus 1. So 99 peptide bonds are there. So 99 water molecule will be released. So 99 into 100 we have to subtract from this molecular weight. So 100 into 110 minus 99 into 18, and that will be our molecular weight of 100 amino acids which are present in a protein. Okay. So the last question is, if molecular weight of glycine is 75, then calculate a circular polymer of 20 glycine. So a circular polymer of 20 glycine and 1 glycine is 75 Dalton, then what will be the molecular weight of that circular polymer of 20 glycine? Now, still now we are seeing that the 5 amino acid, if a peptide have 5 amino acid, then 4 peptide bonds are there. Okay, and if same 5 amino acids are in circular position, in circular form, then we will see how many peptide bonds will be there. There 1, 2, 3, 4 and 5. So in circular polymer for 5 amino acids, so for 5 amino acid there will be 5 peptide bonds. Okay, but in linear form for 5 amino acid there will be 4 peptide bonds. So here for circular polymer for 20 glycine there will be 20 peptide bonds. Okay, and we have to release 20 water molecule. So here 20 into 75, because 75 is the molecular weight of glycine and all total 20 glycines are there, and we have to minus the 20 into 18 because there are 20 peptide bonds are there, so 20 water molecule will be released. So this is the all total molecular weight of a circular polymer containing 20 glycine. Okay, so these are all about the molecular weight calculation, molecular weight of protein and amino acid calculation. Okay, thank you for watching this video.